going to try this. So when we left our time together before snow and viruses invaded, we had started our unit on organelles. So I'm going to pick up where I was when I left. I'll try not to talk with my hands too much. And then we'll try to move through some organelles. Feel free to pause this video any place you want to write anything down, catch the notes. You don't have to, you know, keep up with me. You can always pause and rewind and get your notes down. But I do want you to hear everything I have to say. I know probably very tempting just to zip to where you see the information and write it down and zip to the next one where you see the information. But I'm going to say information too. And it's very helpful for you to hear it, see it, and write it, all three. So here we go. And I'm going to probably be a little clumsy with this because I'm not real great at videos, but I'm going to do my best. So here we go. So we did cell membrane in class. We did cytoplasm in class. We did cell wall in class. Now, sixth period, you've done this next one, Centriole, but the rest of my classes. So if you're not in my sixth period, you need to flip to your notebook page where Centriole, you should have it cut out. This little guy, point down, that guy is waiting for you. So just a brief description of this organelle structure and function. It's very differently structured. Every other organelle we will ever talk about is very blobby shaped and has like membranes, and they're like sacs and bags and blobby noodly shapes. Centrioles are very different. And why they're different and how they came to look so different is still a mystery. There's a lot of research to centrioles because they're not always present in cells. They'll show up when they're needed and they disappear when they're not needed. And they're shaped so oddly compared to other organelles. If you think it looks like a bunch of straws or a bunch of sticks, you're not far off. But their structure is kind of like a Twizzler. I'm not the only teacher that will ever describe them that way. They're important in cell division, clearly. That's what it says. Mitosis, what you guys talked about with WEX, prophase, metaphase, anaphase, and telophase. As the cell gets ready to divide, the chromosomes will get pulled apart. The spindle fibers that reach into the chromosomes at the centromere and rip them apart, those fibers are made by centrioles. You probably drew them when you drew mitosis, and maybe you drew them like Roman numeral 3s or something like that. Pardon me, I'm having a smoothie. It's healthy, right? So it rips the chromosomes apart. That's their main job. That's like when they step onto the stage for their main solo. That's their big job. But they also make spindle fibers. I'm sorry, that is their job. Sorry. And they also make microtubules, which is the last note on here. Microtubules are a lot like the skeleton of a cell. Now, cells obviously don't have bones, but inside the cytoplasm, remember, it's mostly water with some proteins in there. And so what gives the cytoplasm some heft, some gel-like texture is microtubules. They're part of the cytoskeleton, which gives the cytoplasm not so much of a liquid, but something more gel-like or maybe glass-like. We don't know. So I have an image from a microscope. This is a light microscope, not quite an electron scanning microscope. And I'm going to try to use my pen tool. I'm still getting better at this. Okay, so this is a centriole, and this is a centriole. Now, clearly, they don't look the same. They look quite different. The one on the bottom is if you took that drawing that you had, held that bunch of sticks, and looked straight down the middle. So it looks kind of star-shaped, and when it's laid on its side, you can see like the straw-like ones. Here you can see it a little bit better, laying on its side, but these are all centrioles. But because they look so different all the time, now I gotta exit my drawing tool. Yeah, okay. Because they look so different all the time, for like 25 years, biologists thought they were all discovering new organelles. And so in the late 1800s, like 1895, 1898, or something like that, when it kind of got to the end of, wait, our microscopes are getting better, it finally dawned on all these biologists and biologists all over Germany and Austria, England, France, Netherlands. They all thought they discovered a brand new organelle with their improving microscopes, but they all discovered the same one. It's just centrioles were just sitting in different positions. And so, but it turned out to all be the same thing. But it's shaped so differently and so oddly, it didn't really occur to anyone that it was a different organelle, just oriented differently under the microscope. So, wah, wah. But it was officially named the organelle, I wrote it down, in 1895. Organelle, named the Centriole in 1895. But it took like 25 years because people did not understand what they were looking at, which is kind of fun, I think. Anyway, it's a funny little story. All right, ribosomes. So turn the page. Ribosomes are, some teachers make kids like say which organelle is the most important, and they all do extremely important jobs. But the ribosome, when I teach, sorry, I'm having all sorts of word issues. Anyway, so when I teach organelles to kids, I have to teach the importance of proteins. I told you the story about Jesus and proteins in Rishbeater's class, how they are, if you ask yourself anything about a cell, and you can probably answer, like, how does a cell know when it's time to divide? How does a cell know if something is trash? How does a cell know if it needs more of something or less of something or something's attacking it or whatever? You can answer that question primarily by saying, oh, it's a protein. And ribosomes are the organelles that make proteins. So that's their main job. If you write that in your notebook, please put it in all caps, underline it, bold it, highlight it, make it, I don't know, like when you see the ribosome page, I want you to see make proteins as the most important 2 words on the page. However you need to get that done, do it. Ribosomes have a top and a bottom, as you can see in the image. And in between, you've got DNA. Now, I'm skipping a couple steps here. So ribosomes making protein is called protein synthesis. And other than photosynthesis, protein synthesis, I think is the hardest thing, second hardest thing I learned in college. It's really hard. So I'm leaving out a bunch of steps, which kind of makes me feel like I'm lying to you. But I don't want to get bent in too many details you don't need when you're 12 and 13 years old. But essentially, ribosomes read DNA. There's a bunch of steps in between there, but it reads your DNA. You studied DNA before in Wex's class, and maybe even Boggs's class. It looks like a ladder. And so you've got, your 2 bars on the side, and you got your rungs. So it reads 3 rungs at a time. Every 3 rungs, it gets a little code. 3 rungs code. 3 rungs, it's called a codon. So when it reads each of those 3 rungs, it gets some information about how to build a protein. So a section of DNA codes for a protein. Now the section of DNA could be like 100 rungs long, or it could be like 1,000 rungs long. So genes are different shapes and different lengths. But your ribosomes read a section of DNA and make whatever they are told. They do exactly what they are told. They make what they are told. I described Branch to my 6th period when Branch was 4. I made a classic mom mistake. I said, hey, Branch, we're not going anywhere until you get everything off your floor. And I meant put it away. And he said, okay. And he picked everything up and put it on his bed because he was following instructions. Branch was a little ribosome. He does exactly what he was told. And what I got, I wasn't really what I wanted. And a lot of what your ribosomes make isn't exactly what you want, which we'll get to the next organelle in a second, but it will do exactly what it's told. It's going to read whatever your DNA codes for, that's what it makes. So if that DNA is jacked up from like radiation or illness or damage or medication or toxins and your DNA gets damaged, your ribosome will make the wrong protein. And it might be a really crucial protein to your health. Or, so you need your ribosomes to do what they're told. That's good. But if your DNA gets jacked up, they just do what they're told. Okay, ribosomes are, why isn't it coming up? There we go. Sorry. Ribosomes are made in the nucleolus. It is a organelle made in another organelle. The nucleolus we'll get to tomorrow. But the nucleolus is inside the nucleus anyway. But that's where they're made in the nucleolus. Remember, you can hit pause. You don't have to like frantically write down quickly everything I'm saying. Just hit pause. And there are 10 million ribosomes per cell in your body. And you have 30 trillion cells. So you have a lot of ribosomes. I have some pictures to show you of ribosomes underneath the microscope. Remember, you can pause anything. All right, so this is under the microscope. Each of these little gray dots, see if I can go back to my little, no, there we go. My pen tool. Ribosome. I mean, there's probably like, I'm a terrible visual estimator. I don't know. It's not, it's all these dots. All of them. They are all ribosomes. And they are all following instructions, spitting out proteins all day long. That is on the rough ER, which we'll get to in just a second. But yeah, these are all ribosomes. And this is like a tiny little section of your rough ER, and there's so many ribosomes. Even these little like whispers of gray in the back that are probably ribosomes, they're ribosomes. They're all ribosomes. 10 million per cell in your body. A lot. All right, so this is a protein. This is a protein you may have heard of when you studied the circulatory system in Mr. Wexler's class. The protein you're looking at on your screen is hemoglobin. Hemoglobin helps your red blood cells carry oxygen. Maybe Wexler talked about that. But it is a protein. It's made by ribosomes. And it is very typical of a protein shape. I like to describe proteins as looking like broken slinkies, mess up hairballs. I don't know. But as much as this thing looks like a mess, I promise if it was not in the shape it was in, it wouldn't work. Now, the important part about a hemoglobin shape is it's open in the middle. That section in the middle that makes it look kind of like a donut, this donut hole, really, is where the oxygen sits. It's reactive to oxygen, so oxygen is held in there, and it will carry the oxygen to other cells in your body that need it. So that's hemoglobin. I have another protein to show you. Isn't this fun? Okay, so there's actually something going on in this picture you might be familiar with. Throughout the middle of the picture, it looks like very thin neon green kind of chains. Those are lipids. And if you look at the top and the bottom, you'll see their little heads. And you're like, that's not how I drew it for you, Miss Sword. You're right. You want me to draw? You want to draw that? Or you want to draw your like people with the two legs? You want to draw the people. I know you do. So those are all lipids. And all these hairball things in the middle that look like curlicue old school phone cords, those are all proteins. So the other day, when I told you about Jesus and proteins on your cell membrane day, we just drew like big kind of beans stuck in the cell membrane. Those beans represent these hairball looking things. These are proteins. This is a real protein. And while it looks like a mess, I want you to pick any of these proteins that you're looking at right now on your screen. The peach ones, the teal ones, the green ones, the pink ones, the blue ones, I don't care. Just look at any of them outside the cell, which are on the top, and then you'll see that they're like in or out. So if it's faced in, if for some other reason, if the ribosome was messed up and kinked that protein the opposite direction, that protein would not work. So even though it looks like a mess and looks like chaos, every curve, every bend, every twist to these proteins is purposeful and necessary. And it looks crazy, but it's the way they work. They're like keys. They work like keys and locks. But they have to be just the right shape so they'll go in. Okay. Golgi bodies. Wait, I'm going to hit pause. Hold on. We'll come back to Golgi in just a second.